DNA replication is the process whereby an entire double-stranded DNA is copied to produce a second identical DNA double helix. The objectives of this exercise are to understand the functions of the proteins responsible for DNA replication, including helicase, SSB protein, primase, the sliding clamp, DNA polymerase, RNase H, and DNA ligase, and to understand why the leading strand is synthesized continuously and the lagging strand is synthesized discontinuously. DNA replication is an intricate process requiring the concerted action of many different proteins. The replication proteins are clustered together in particular locations in the cell and may therefore be regarded as a small replication factory that manufactures DNA copies. The DNA to be copied is fed through the factory, much as a reel of film is fed through a movie projector. The incoming DNA double helix is split into two single strands, and each original single strand becomes half of a new DNA double helix. Because each resulting DNA double helix retains one strand of the original DNA, DNA replication is said to be semi-conservative. DNA replication requires a variety of proteins. Each protein performs a specific function in the production of the new DNA strands. Helicase, made of 6 proteins arranged in a ring shape, unwinds the DNA double helix into 2 individual strands. Single-strand binding proteins, or SSBs, are tetramers that coat the single-stranded DNA. This prevents the DNA strands from reannealing to form double-stranded DNA. Primase is an RNA polymerase that synthesizes the short RNA primers needed to start the strand replication process. DNA polymerase is a hand-shaped enzyme that strings nucleotides together to form a DNA strand. The sliding clamp is an accessory protein that helps hold the DNA polymerase onto the DNA strand during replication. RNase H removes the RNA primers that previously began the DNA strand synthesis. DNA ligase links short stretches of DNA together to create one long, continuous DNA strand. Let's look at the steps of DNA replication in more detail. To begin the process of DNA replication, the 2 double helix strands are unwound and separated from each other by the helicase enzyme. The point where the DNA is separated into single strands and where new DNA will be synthesized is known as the replication fork. Single-strand binding proteins, or SSBs, quickly coat the newly exposed single strands. SSBs maintain the separated strands during DNA replication. Without the SSBs, the complementary DNA strands could easily snap back together. SSBs bind loosely to the DNA and are displaced when the polymerase enzymes begin synthesizing the new DNA strands. Now that they are separated, the 2 single DNA strands can act as templates for the production of 2 new complementary DNA strands. Remember that the double helix consists of 2 anti-parallel DNA strands with complementary 5' to 3' strands running in opposite directions. Polymerase enzymes can synthesize nucleic acid strands only in the 5' to 3' direction, hooking the 5' phosphate group of an incoming nucleotide onto the 3' hydroxyl group at the end of the growing nucleic acid chain. Because the chain grows by extension off the 3' hydroxyl group,